Somshekhar?
Yeah, Dr. Rakesh. So, very good afternoon to all.
Department of Botany alone. Voice.
Actually, Department of Botany allow ಮಾಡಬೇಕಂತ ಕೇಳಿಸ್ತಾ ಇಲ್ಲ ವಾಯ್ಸ್.
Sorry. Let me do that. What is it called? Department of Botany allow. Yes. Maybe Department of Botany I will just make as panelist. So yeah.
Yeah, a very good afternoon to all. Am I audible?
Yes.
Yeah. So I'm very glad to invite you all for the webinar conducted by the IDST Government College, Chikmagalur, on the occasion of 70th anniversary celebration of the college. On behalf of principal of the college and the coordinator of IQAC and the Department of Botany, I heartily welcome you all for this webinar. So this webinar is conducted by this IDSG Government College situated in Chikmagalur. So this Chikmagalur is situated in the Western Ghat region of Karnataka. And this Chikmagalur is the place where the first coffee cultivation happened in India. And about the, I'm very glad to give a brief introduction regarding our college. So this college is celebrating the Vajra Mahotsava, that is the 75th year of establishment. And I am very glad that I am the alumni of this college. And this college is affiliated to Coimbatore University, and it has NAC A grade as well. And it caters the students from a rural background. In the college, both UG and PG courses are being run. So presently, there are around 3,000 students are studying in this college. So in our botany department also, we have both the UG and PG courses. Presently, there are around 200 students.
I think Somshekhar, you're on mute. Hello.
Yeah, Rakesh.
So sorry, I think you were on mute for a couple of seconds. Sorry.
Okay, shall I continue then?
Yes.
Yeah. So in our botany department, we have both UG and PG courses. Presently, there are around 200 students which are studying in our department. So it caters for both UG and PG students. Okay. And once again, I welcome all the participants for this webinar. So let me give brief glimpses on the webinar. So being a human, we can cope up with any kind of adverse environmental condition. It's maybe with respect to the temperature as well. If the temperature is increased, we have different strategies to cope up with that. So we can escape from that habit, or we have the fans and ACs and coolers. There's so many things, but that is not the case for the plants. So they need to complete their life cycle. They need to cope up with any kind of conditions where the seed is get germinated. They can't escape the conditions. But during this era, we know that global warming affect all the organisms, including plants. So the increasing temperature may affect the overall growth of the plant, including photosynthesis. So interestingly, Dr. Rakesh, the present day speaker, is working on how the plants are cope up with the increasing the temperature, how that photosynthesis is going to be affected. So he's working on that wonderful topic. And let me take this an opportunity to introduce Mr. Rakesh. So Dr. Rakesh is from Karnataka, basically from Shimoga, Karnataka. He completed his undergraduate in Shimoga and post-graduation from Department of Applied Botany, Coimbatore University with 2 gold medals. So later he moved to IASC Bangalore. Then he worked as a low carbon farming expert at Environmental Defense Fund. So for many years. Then he moved to Leeds for his doctoral degree. So he obtained his doctoral degree from Leeds University under the supervision of David Galbraith. And after the completion of his doctoral degree, he moved to Leeds University for his postdoctoral studies under the supervision of Emmanuel Glure. And he completed one more postdoc at Birmingham University under the supervision of Christian Foyer. So presently, he is awardee of prestigious Werner Gren Fellow at Uppsala University, Sweden. Sweden, under the supervision of Robert Mascarella. So presently, he is working on the thermal tolerance of the plants with respect to the photosynthesis, and he is conducting his research in Amazon, Puerto Rico, and Western Ghats in India. So presently, his research is being conducted in Amazon, Puerto Rico, and Western Ghats in India. So yes, so let us start the session. Now I am hand over to Rakesh Tiwari. So after the session, we have a discussion session as well. After that, our principal madam will give a small glimpses on the webinar, and the whole program is now being continued by Ganesh Shinde sir, is the HOD of our department. Now it's to the Rakesh. All of you can enjoy his talk, including some demonstration for the how to measure the photosynthesis. So thank you, Rakesh. It's now your time to continue the session.
Thank you, Somshekhar. Can you guys hear me? And I think
sir, your camera is not visible. You are not visible.
can you see my slides?
No.
Huh. Okay. Video. Can you see now?
No.
Can you try different? I think it can.
My phone it is coming.
So there are different screen, I suppose. So there is my video. There is, I think there is going to be a slide which you can see. Can you check, please? And we have about 46 people attending online. So I want to just check if people are able to hear me, hear you guys.
Yeah, I can see you.
Hi, Evan.
Hi, Rakesh. I can see you. I can see you both yourself as well as on your screen also available.
So the slide, you can see the slides as well, right?
Slides, yes.
Okay. So if the team in the seminar hall can actually try and swap the screen to the slides, so we can start.
One screen.
Yeah, sorry about this. So usually these things are quite common, and we didn't manage to test this before, so I apologize. So I think we should be able to start in a couple of minutes.
Now it is visible. You can start, sir.
All right, great. Thank you, everyone, and thank you for the opportunity, and thank you, Somshekar, for the introduction. Would it be possible if I actually, wait, just 1 second. How do I make this bigger? Just 1 second. I was just trying to see. Okay, so so thank you for this opportunity. And then in this quick introduction, what Somshekar did not mention was that we are friends since 2004. So we studied together. We were in the same university, Kuwait University, and then that's we are still connected and we are still working together. And that's how we are now working. And I also would like to request if we can unmute the students who are sitting in the hall so that this can be an interactive session, right? So the title of the topics, as I decided, was measuring heat sensitivity of photosynthesis in plants. So let's just stick to that title now. And if anybody has managed to scan this QR, so we'll know a bit about you, but this is not important. Now, let me go to the very first slide. Okay. Now, so what do we plan to achieve? So we want to actually describe how do we measure photosynthesis and its heat sensitivity. This is mainly meant for undergraduate students. And this is not going to be an advanced, okay, sorry, just 1 second. If you can you guys see the slides or not yet? Okay. Hello. Can you guys see my slide? Sorry.
Actually, no, sir.
No. So I just restarted. So let me just get it again. I'm so sorry about this. Share. It should be this one. Share. Right. Okay. So now you should be able to see the screen. Can the students see the slide now?
Yes, sir.
Okay. So maybe if we can sort of swap the students and then so that I can start interacting with them. So this is more of an interactive session rather than a lecture session. So if I can get the students in the front, right, if that is possible.
Video is seen, no, sir.
Yeah. So I can see you. I think this is it Ganesh?
Students are no, yeah.
Yeah. So if I think students will not be able to respond while you are in the front. So if the students can actually come to the front, so whoever wants to learn more can come to the front. So now I feel like I'm actually in the classroom. So I want to talk and then okay, just one person. No more. Don't worry, there'll be no examinations, nothing. So we'll just learn. We'll try and see what we understand, and we'll try and learn a little bit more than what we know. Okay, so please use your voices. So you can probably see a little window of what I can see, but I can hear your voices. So did you guys have your lunch already? Oh dear God, maybe no. ಊಟ ಆಯ್ತೇನ್ರಿ ಎಲ್ರದ್ದು? So okay, so do you guys understand English or do you want this in Kannada? Because I think there are audience online who are probably not who cannot speak Kannada. We can have question and answer sessions in Kannada particularly, but can we have this in English, or should I make my accent a bit less Indian, a bit more Indian? Ganeshji, can you please help?
Yeah, I can. Martin, sir.
So what I meant is, okay, let me just do this bit in Kannada. So ಎಲ್ರಿಗೂ ನಾನು ಹೇಳ್ತಾ ಇರೋ ಇಂಗ್ಲಿಷ್ ಅರ್ಥವಾಗ್ತಿದ್ಯಾ? ಓಕೆನಾ? ಸ್ವಲ್ಪ ರೆಸ್ಪಾಂಡ್ ಮಾಡ್ತಾ ಹೋಗಿ, ಯಾಕಂದ್ರೆ ಈ ಟಾಪಿಕ್ ಏನಿದೆಯಲ್ಲ, ನೀವು ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ಅಲ್ಲಿರೋ ಹಾಗೆ ಕಲಿತಾ ಹೋಗ್ಬಿಡ್ಬೋದು, ಬಟ್ ಮೆಷರ್ ಹೇಗೆ ಮಾಡೋದು ಅನ್ನೋದನ್ನ ಸ್ವಲ್ಪ ಇಂಟರಾಕ್ಟಿವ್ ಆದ್ರೆ ಎಲ್ರಿಗೂ ಅರ್ಥ ಆಗುತ್ತೆ. ಸೊ ಉತ್ತರ ಕೊಡ್ತಾ ಹೋಗಿ. ಸ್ವಲ್ಪ ಊಟ ಆಗಿದೆ ತಾನೆ ಎಲ್ರದ್ದು? ಸೊ ಏನಾದ್ರು ಮಧ್ಯದಲ್ಲಿ ಸ್ಲೋ ಆಯ್ತು ಅಥವಾ ಫಾಸ್ಟ್ ಆಯ್ತು ಅಂದ್ರೆ ಹೇಳಿ, ನಿಲ್ಲಿಸಿ, ನಿಧಾನ ಮಾಡಿ. ಓಕೆನಾ? ಸೊ let us see what we want to achieve in this session. So I would like to describe how we measure. I think there are more people signed up online in the same rooms. That's why you're hearing the echo. So whoever is joining online, please mute their mic in that room, in that seminar room or lab. Right? Okay. So, and the second aspect is we would see if we can share some results from Western Ghats and probably Amazonian forest. But this will all depend on how we interact and how much we learn. Okay. So the outline of this talk is very simple. We will just touch upon what happens during photosynthesis, how to measure photosynthesis, and in particular, how to measure temperature sensitivity of photosynthesis. Okay. Somebody is raising the hand. Just a second. Let me see. Not sure. Okay, so just in the interest of time, since I'm not able to figure this out across two monitors, I'll just go ahead. Students, click ಹೇಳ್ತಾ ಇದೆಯಲ್ವಾ? Screen ಕಾಣಿಸ್ತಾ ಇದೆಯಲ್ವಾ? Okay.
Yes, sir.
Let's go ahead. Okay. So we will talk about mainly two measures of temperature sensitivity of photosynthesis and how we do it. And then we will also describe what we measured in the Western Ghats forest. Okay. Now, what happens during photosynthesis? Can one of you guys just throw some points as to what we know about photosynthesis in higher plants? Where does photosynthesis happen? Leaves. So, which animals do photosynthesize? Do animals photosynthesize? Yes? No? Do you know any animal that photosynthesizes? No? Okay. I need some responses, or else we'll go into more complicated topics as we go. So don't hesitate. Nobody will grade. Nobody will give you marks on what you respond now. So you'll only learn. Do you know any animal that does photosynthesis? No. So it is only plants that do photosynthesis, right? So plants, thinking of terrestrial plants, every plant that you see around you are photosynthesizing. Do you also know that there are photosynthesizing organisms in the ocean? Right? So they also photosynthesize, they also capture carbon dioxide from the atmosphere, right? And there are also bacteria, cyanobacteria, and single-celled organisms that do photosynthesis. So in this particular session, we will only talk about photosynthesis among higher plants, which is terrestrial plants. Okay, not aquatic organisms or not the microbial photosynthesis, right? What happens during photosynthesis? Do you recognize any of this? What do you see here? This is like a very bare minimum description of photosynthesis. One part of the photosynthesis. So what is happening here? The primary ingredient, the energy source, which is sunlight. Light, of course, you can use all your artificial light, but mainly on planet Earth, what is the source of energy for photosynthesis on our planet? That's sunlight, right? So, and what is the other ingredient? You already see here. This is a simple This is water. We need water for photosynthesis. Do we need water or no?
Yes, sir.
Why do we need water? What happens to water during photosynthesis?
For the purpose of electron transfer and
Okay, we will transfer electrons from where to where. Good, good.
so from photosystem 2 to 1.
Okay, so why do we care about photosystem 2? Why do we care about photosystem 1? What happens? Why do the electrons need to travel?
Photons and some
The
movement of the
photoreceptors.
photons takes place. Photosystem 2 to 1.
Okay, okay. So what you see here, this is a representation of photosystem 2 and a bit of light harvesting complex. So there are these antenna complexes, photosystem itself. There are different proteins inside that hold different components, right, which are arranged in different order of their redox potential, and they absorb light. In fact, photosystem 2 directly does not absorb light so much. It is the light harvesting complexes. They channel that energy into the photosystem, into the reaction center. You know what reaction center's role is? What does reaction center do?
Chlorophyll B. Chlorophyll A. Sorry.
No, that's okay. So you will find chlorophyll A, chlorophyll B, carotenoids, xanthophyll carotenoids, everything in these photosystems. But what is photosynthetic reaction center? Just in a simple way, what happens here? This is the most exciting component on planet Earth for lifespan. Without this, no life exists, no aerobic organisms at all on this planet. We are breathing oxygen, right? So 21% of our atmosphere is oxygen. How does that oxygen, how was that produced over millions of years on this planet, enabling evolution of terrestrial organisms and all the other forms? It is in the photosynthetic reaction center of various lower forms that oxygen was produced, and it is getting produced all the time. So the role of photosynthetic reaction center in photosystem 2 is to split water and generate oxygen, free radicals, and protons. So it produces H plus and O, and then eventually it produces molecular oxygen, right? And that is what is released during photosynthesis. That is what higher organisms use for their respiration process, right? I can hear my own voice. Maybe someone is logged in on a small computer or a phone. Please, could you mute your mic on that room? Thank you. So now, so photosynthetic reaction center where water is split, proton is generated. But where are these photosystems located? Can you guys tell me? Where are they located? Where do you find them?
Thylakoid
Thylakoid membrane. Where are those thylakoid membranes?
It's in a chloroplast.
In the chloroplast, right? And where are those chloroplasts?
It's
In the leaves, generally for higher plants, we're talking about leaves. So when we look at a cross section of the leaf, we have different layers of cells, palisade parenchyma, spongy parenchyma, right? And in the cytoplasm, there are these chloroplasts all over. And those chloroplasts, if you zoom in, they have membrane, thylakoid membranes. And on these membranes, you'll find a lot of these pigment protein complexes, and one among them is photosynthesis. So there are other pigment protein complexes, they have different roles in photosynthesis. This is a oversimplified representation. So let's go forward. So with this, we also generate ATP and NADPH across due to the proton motive force, and proton is generated by splitting water and by using energy from the sun, right? And then that ATP and NADPH is used to capture carbon dioxide, right? To sequester, to uptake carbon dioxide. And I wanted to ask what is the most abundant protein on this planet, but this is a stupid slide. I've already mentioned the answer. So, do you know what is Rubis program?
Enzyme.
Rubisco is an enzyme, and what does it do?
Faster.
Captures oxygen or carbon dioxide capture.
Yes, it binds with oxygen and carbon dioxide. Yes, very good. And then what is the first part of that name? What does that represent? So there are Rubisco, right? So there are two parts. What does the Rubis and CO represent? So you explained CO. What does Rubis mean?
So rubulose bis phosphate.
Excellent, right? So it actually describes what substrate it binds with. It binds with ribulose, right? And then it can also bind with carbon dioxide. There are different binding sites. And that carbon is unstable. It splits into 2,3-carbon stable compound. And that is why generally most organisms, terrestrial organisms, do C3 photosynthesis. So we will not go into C3, C4 and stuff. We will just talk about the main enzyme, which is Rubisco, right? This is the enzyme that captures or attaches carbon dioxide and then attaches it to a 5-carbon sugar, leading to sequestration and the dark reaction or the Kelvin cycle or Kelvin-Benson-Baschen cycle. There are different names. Basically, a way of capturing carbon and then reproducing RUBP for the next step, right? So these are the two main components, or the two main processes happening during photosynthesis, right? Um, and if we were to measure this, what can we measure? If we have to measure photosynthesis, what all components can we measure from this? If you see the schematic, what comes to mind? How do we measure this? What are the signals that we can get from this process represented in the screen?
First change ಆಗ್ತಿಲ್ಲ sir.
Changes right.
Changes in, yes.
Slide changes. Slide.
Sir, we are not able to see the changes in the slide change. Still, we are in objective slide.
Oh dear God. Okay, wow. So can the online audience see the slides? Okay, so let me just stop sharing. I think can the online audience, could somebody respond and tell me if you are able to see the slides? I can stop sharing and then I can start sharing again. That. So now you should be able to see this screen. Can you see it now?
Yes, sir.
Okay, sorry about that. So, and then if you don't see the slides go or move, just let me know. I will tell you when I'm moving to the next slide. Okay. So right. So now what components do you see here? So this is the process that we spoke about. There is sunlight, and it's absorbed by photosystem 2, photosystem 1, and all of them. But mainly here we represent photosystem 2. That's where the reaction center is, splits water, generates proton, also produces oxygen, and in the process leads to generation of energy and reduction power for carbon dioxide capture. And then we have the Rubisco enzyme binds with carbon dioxide that also binds with oxygen, and it binds with the substrate. What is the substrate for Rubisco? I can't hear you guys. You have to speak out louder, please. Make a lot of noise. Don't worry. What is what substrates do Rubisco bind with? It binds with ribulose, right? So the 5-carbon sugar, it binds and then it generates 3-carbon stable compounds, and that's how the carbon dioxide capture happens. So from this slide, can you tell me what changes are occurring in the leaf that we can try and measure? what is getting up in this process? Carbon dioxide, right? So can you measure CO2 uptake rates, right? I'm not sure if I can see the slides. The students are very quiet now somehow.
Photosystem to liberate the oxygen.
So you have oxygen evolution, so you can actually try and measure how much oxygen is produced in the leaf, right? That is one way of measuring it. The other way of measuring it is to see how much carbon dioxide is absorbed, correct? Right. So can someone tell me what is the concentration of oxygen in the atmosphere? Can you guys tell me the concentration of oxygen in the atmosphere? 21%, right? And then what is the concentration of carbon dioxide?
0.04.
How much?
0.01.
0.04. So it's actually 420 plus ppm, parts per million volume, or if you put it in percentage level, 0.04. So imagine 21% and 0.04%. It's very, very tiny concentration of carbon dioxide compared to oxygen. So in terrestrial plants, if you were to measure changes in oxygen, which is tiny proportion change compared to the total concentration in the atmosphere, it will be very difficult for terrestrial organisms. For example, if you want to measure it in aquatic organisms, what we measure there is dissolved oxygen that is generated by the leaves that you can measure in the water. So you can measure dissolved oxygen changes, and that will give you oxygen emission rates. Okay. Now, the only reasonable way to measure photosynthesis rate in higher plants, the most reliable one, is to measure carbon dioxide uptake rates. Is that okay? Shall we go to the next slide? Okay. So now there are a couple of instruments that you can use to measure these. They look very fancy, right? So I've represented one kind of instrument here, another kind of instrument here, but they look very complicated. But in essence, they are very simple. Okay. Now, let's first look at how to measure carbon dioxide. And what do you see here? So this is the picture taken in Sirsi, in the Western Ghats forest. And if you observe here, I don't know if you can see the cursor, but there is a leaf clipped into this camera-like looking instrument, right? Have you seen this before? No, right? And this is an instrument to measure carbon dioxide uptake rate, and we will understand how to do it and how this instrument works. Okay. Now let's go back to your a bit of a physics class. Okay. Do you remember this slide? You see an electromagnetic spectrum with different parts identified. You see the slide change. Okay, can some student respond? Have you seen the slides changing? Guys, do you see the slide change? I don't know if you can hear me. What do you see in the slide?
Wavelength of the light.
Sorry, speak up loud, please.
Wavelength of the light.
Yes, so you see a representation of an electromagnetic spectrum, which represents different energy levels of electromagnetic part of the light coming into the Earth's atmosphere, right? So this part, about 400 to 700 nanometer, is our visible part of the spectrum. We have different colors which represent different energy of the light, and the higher the wavelength, the energy goes down, the lower the wavelength, the energy goes up. So these are high energy radiation or electromagnetic radiation. For example, you see ultraviolet here, which can damage. But then when you do blue light, for example, if you consider this section, they're very high energy. Green, not so much. And if you come towards red, they are low energy light. Okay. And if you go beyond, this is the light our eyes, human eyes, cannot see, right? But this is a part of the spectrum, the shortwave infrared and the thermal infrared spectrum, where vibrational energy of molecules are can be detected. So when you rub your hand, it's vibration energy, right? So you generate heat. Similarly, when molecules are heated, the vibration energy can be seen in this part of the spectrum. So I just want you to look at this part, where we are going to detect carbon dioxide. Okay. Now, when you measure what is coming into the Earth's atmosphere, so on the Earth there's a layer of air, right? And if you go outside the atmosphere, if you're looking at, for example, if you're sitting on a satellite and you measure what is coming from the sun, you get something like this yellow spectrum. It doesn't have any absorption lines. You see the yellow lines here, which is also represented by curve. Do you see this? Hmm? So, and this is the part of the light that we see with our eyes. And what you also see here, you see this much more distorted lines here. These part of the spectrum, right? This is the spectra of when we are on the Earth's surface. When the sunlight is passing through the atmosphere, some of it is absorbed, and the absorption is based on what is in the atmosphere. And you can see that water has different absorption regions here. Carbon dioxide also has an absorption region here at around 2000 nanometer. You see this one, right? So this means if you shine infrared light on carbon dioxide, it absorbs. Do we understand this at this point? Can some students respond, or shall I just go ahead, because it's going to be a bit more complicated as we go. I need some response. So, do you guys understand until here? Yeah. So, can we see the light that carbon dioxide absorbs? You think our eyes can see the light that carbon dioxide absorbs in the infrared part of the spectrum?
Sir, we could not able to hear your voice clearly, sir.
Okay. Can you hear me now? Can you hear me now?
Yes.
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 Okay. So when you look at this, the absorption lines for carbon dioxide is at 2000 nanometers, right? Do you think we can see that part of the spectrum? Can we see that light? If I have an infrared lamp, infrared source, can you see that light with your eyes? What do you think?
With instrument.
With instrument, we can see all of it. We can have the full sensors. But can we see it with our eyes? No, exactly. So yeah, please start responding, because it will become very slow and very boring if you don't respond. So if I don't know what was different earlier, you guys are responding quite well. ಮುಂಚೆ respond ಮಾಡ್ತಾ ಇದ್ರಿ, ಇವಾಗ respond ಮಾಡ್ತಾ ಇಲ್ಲ. There is something that can change. Please do it, because this is going to be not so useful if you don't respond. Okay, I can just give a lecture and then go forward. Nobody will understand this. Okay, it's mainly for these students, and if you respond, I will try and make you understand this better, hopefully. Okay, so now we know that in the infrared part of the spectrum, carbon dioxide absorbs light. Okay, now can we use this to measure carbon dioxide concentration? Do you see a slide change? Can somebody describe what is on the slide? If not,
Infrared.
Describe in Kannada, you can do it.
So there is an infrared light source.
Right.
So and there is a cabin which contains carbon dioxide.
Yes.
Next to it is a infrared light sensor.
Right. Right. So this is like you have a source and you have a detector. So I've labeled it as S and D. Right. So what do you think these carbon dioxide molecules are doing? So they're absorbing light. So let us say we send 100 units of energy to the lamp. How much energy will be detected? Will it be 100 or less or more? Hmm. So what do you think? So if carbon dioxide absorbs infrared light, and if we send 100 units of infrared energy into this chamber, and after it absorbs, we check how much is detected on this sensor. Will it be 100 or less or more?
Less.
Yes, exactly. Yes. Now, consider the situation where in this chamber there is no carbon dioxide. Okay. So, whatever, if I send 100 units of infrared light, how much will we get here? 100, right? So, this is it. Likewise, if we increase the concentration of carbon dioxide, the amount of energy that we detect here will be less, right? So this is the idea of infrared gas analyzers. Did you guys get it? Can someone explain this to me?
Yes, sir. Sure.
Yes, tell me. This is the most complicated slide in this entire presentation, so you don't have to worry. So if someone can explain me what this is a little bit in Kannada or English, whatever you guys prefer, we can go ahead. After this, it will be simple. If we understand this concept.
So this the amount of the equivalent device sensor minus ಮಾಡಿದ್ರೆ the amount of concentration carbon dioxide ಗೊತ್ತಾಗುತ್ತೆ equal to the concentration of carbon dioxide.
Can you ಸ್ವಲ್ಪ ಮೈಕ್ ಹತ್ರ ಬಂದು ಮಾತಾಡ್ತೀರಾ? I think you're explaining it right. Could you come close to the screen I'm viewing and then tell me? Yes.
Sir, the source ಅಲ್ಲಿ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಇದು ಇನ್ಫ್ರಾರೆಡ್ ರೇ ರಿಲೀಸ್ ಆದಾಗ ಅಮೌಂಟ್ ಆಫ್ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಪ್ರೆಸೆಂಟ್ ಇರುತ್ತೆ ಬಾಕ್ಸ್ ಒಳಗೆ ಕಂಟೈನರ್ ಒಳಗೆ ದಟ್ ಈಸ್ ಅಂದ್ರೆ ಎಷ್ಟು ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಇನ್ಫ್ರಾರೆಡ್ ರೇ ಅಬ್ಸರ್ವ್ ಆಗಿದೆ that is equal to the amount of carbon dioxide present in the box. ಅಂದ್ರೆ ಸೆನ್ಸರ್ ಅಲ್ಲಿ ಎಷ್ಟು ಕಲೆಕ್ಟ್ ಆಗುತ್ತೆ ಅದು ಅಮೌಂಟ್ ಆಗಿರುತ್ತೆ.
Okay. So
proportional
yes. So if the concentration of carbon dioxide increases here, the amount of light detected in the detector will decrease or increase. Reduce, exactly. So that's so what we do is we calibrate the system and we call this as infrared greenhouse gas analyzer, or we call it infrared gas analyzer. Why do we call this infrared? Because the molecules absorb in the infrared part of the spectrum. We have an infrared source. Okay, it is analyzing gas. Okay, so that's why you can call this as infrared gas analyzer. You can also call it infrared greenhouse gas analyzer. Why do we call this greenhouse gas analyzer?
They are responsible for the increase of heat.
Any gas that absorbs thermal energy, heat, and holds it for a long period, that gas is considered a greenhouse gas, right? Can you tell me other greenhouse gases that you know of?
Carbon monoxide.
Okay.
Monoxides. All monoxides.
Okay.
Water vapor.
Exactly. Water vapor is a greenhouse gas. And you know one interesting thing about this, the water absorption lines are actually very close to the carbon dioxide absorption line. There are multiple absorption lines that we have seen in the spectrum. Okay. So what the photosynthesis analyzer have is they have a light source, they have a sensor which can detect just the area where carbon dioxide absorbs, and another sensor which can detect just the area where water absorbs. Okay, so there is one source, maybe there are two detectors. They are called photodiodes. They're just simple diodes that detect energy of a particular waveband based on the filter the material they have. So this is the principle of infrared gas analyzer. Okay. Now imagine if you have this system. Let us say you have this third situation. What happens if we put a leaf inside here?
Amount of carbon dioxide is reduced.
Exactly. Why?
Photosynthesis. Photosynthesis. Photosynthesis.
Photosynthesis. We will not touch upon photorespiration today. Respiration is even more complicated. Okay. So
Carbon dioxide
since it is
wasting.
exactly. So since it's absorbing carbon dioxide, the amount of carbon dioxide molecules will decline in the chamber, right? And the amount of light detected here will increase or decrease?
Increases.
Increases. Excellent. That's all. That is the whole idea of this infrared gas analyzer. Okay. So similarly, what you see here is we have two of these. So think of this as one analyzer, another analyzer. Okay. And in between, you have a chamber where you can keep your leaf, which is photosynthesizing. Okay. So you send a known amount of carbon dioxide into this. You measure what is absorbed and what is detected here. Sorry. And after it interacts with the leaf, you detect again. So if the leaf here is photosynthesizing, this and this difference in the two erga will give you the rate of change. Is that reasonable? Does it make sense? Yeah. I can see some questions. So if you have any questions, tell me. So let's put some numbers here.
Excuse me, sir.
Yes.
Sir, can you repeat that because there was a little bit disturbance between.
Okay. So we understood this one, right? So this slide, right? Now, imagine we put one of that erga here, another erga here, okay? And then in between these two, we have a leaf, okay? So here, what is the concentration of carbon dioxide in the atmosphere?
Reduces.
No, what is the carbon dioxide concentration in the Earth's atmosphere right now? 420 ppm or something like that, right? So 0.04% or 400 ppm, correct?
Yes.
So we are allowing a flow of that air through here, which has, let us say, 400 ppm carbon dioxide. Okay, and it passes through the leaf, and then we detect here. What would you expect here if the leaf is photosynthesizing? Will it be 400 ppm or less than 400 ppm?
Less than 400.
Exactly. So that is what happens here. So we have two units of erga, infrared gas analyzer. Okay, one before the leaf chamber and one after the leaf chamber. The difference between these two, the reference and the sample erga, will give you the rate of change of CO2 uptake. Does it make sense? So when you say rate of change, do you need a time component there per unit time, right? So you need either seconds or minutes, hours, or days, correct? So when you talk about the rate of any component, meters per second, right? So if you're talking about the displacement, so there is a time, there's a distance, there is a time component. So here there is also a unit called carbon dioxide per unit area of leaf per second. Does it make sense? Okay. So now let's go and look at how this is like combined. So you saw that instrument, something like this. You saw it like this, right? So all of this internally looks like this. So you have a gas plumbing where it takes it to the reference erga and sample erga, and in between these two, there is this leaf chamber where you can expose it and then measure photosynthesis on that area of the leaf. Is it okay? Does it make sense? Don't worry about the tubing and stuff. So all you need to know is this. This you have understood, right? This is represented here in a very different direction. But what you need to see is, do you see a leaf cuvette here? You see leaf cuvette? You know what's a cuvette? Cuvette, whatever you want to call it. What is a cuvette?
Glass.
Something that closes like this, right? So you have in this instrument where you can clip the leaf. Can you see the slide changing? You see this instrument clipping onto the leaf, right? Or shall we go to another slide and show you something? Okay, so here, I think here you can see it a little bit more. Can you see another instrument where the leaf is inside the chamber, right? So what is happening here is a fixed area of the leaf getting measured, and that area is important to calculate the photosynthesis rate as well, because all the photosynthesis units of photosynthesis, as we know, will be in micromoles of carbon dioxide per meter square or centimeter square, however you represent, per second. So there is the gas, there is the area, there is the time. So we know that we count time in the instrument. We need to account for the leaf area that we are measuring here in the cuvette. And then the ERGA will give you the concentration after the calibration of the instrument. Is that okay? Do we understand it until here? Any questions? So let's have some questions from the students. So we move forward a little bit. And any questions? Any understand anything in this?
Last page, content page like this.
Any
Like this.
No, okay. So let me just move on. I think, okay. So in this instrument, what is also additionally important is that you can control everything that is to do with the leaf environment. So what affects photosynthesis? Okay, let's just have a simple chat. What affects photosynthesis? What environmental parameters affect photosynthesis? ಸ್ಥಿತಿ ಸಂಶ್ಲೇಷಣೆ ಕ್ರಿಯೆಯನ್ನ ಯಾವ ಯಾವ ವಾತಾವರಣದ ವಿಷಯಗಳು ಪ್ರಭಾವಿಸುತ್ತವೆ? ಕನ್ನಡ ಬಳಸ್ತೇನೆ. I need some answers.
Sunlight.
Sunlight, right? If you have low light levels on the leaf, photosynthesis rate will be
Decrease.
which will be low, okay? And if you increase the amount of light, what happens? Photosynthesis will increase, and then at some point it will saturate, right? So because the total amount of light that the photosystems and everything can absorb and what they can do with it will saturate. So you will find a curve which will plateau like this. Okay, now let us say carbon dioxide concentration. What happens if you change carbon dioxide concentration around the leaf?
Sure. If it is high, the process will go with C3 cycle. If the concentration of carbon dioxide is low, it goes for photorespiration.
No, not really. Okay, so the proportion of photorespiration changes at different levels. So let's not talk about photorespiration. Let's just simply try and understand what happens when you change carbon dioxide around the leaf. So you see this situation, right? This figure here. The concentration of carbon dioxide is 420 ppm, right? What if I reduce it to 200?
Repeat the question.
So the concentration of carbon dioxide in our atmosphere is 420 ppm, right? What if I reduce the carbon dioxide concentration to 200 ppm, half? Photosynthesis rate will increase or decrease?
Decreases.
Decrease. What if I reduce it to 50? Decreases. What if I increase it to more than 400? Let's say we go to 500.
Increase.
Increases. So similar to the light response,
Somebody taken.
if you change the
Maybe it is
carbon dioxide concentration from 0 to 500, 1000, 2000 micromoles per mole, you will see a saturation curve like this. I did not put any of those curves because I didn't want to go into details. We can have it in the next session, for example, right? So you also find an increasing part where with increasing carbon dioxide, you will find increasing
printer
rate, but it saturates at some point. Okay. Now, what will happen when you increase the temperature of the leaf? Anybody? Because that is our topic, right? So we want to understand heat sensitivity of photosynthesis. So what is the temperature of the room right now?
This I'm giving to finance color printout. Just knock the door and ask her to print and take.
I can hear color printout and stuff. Can somebody mute their mic? Thank you. Now, let's think about what will happen if we change the temperature of the leaf.
So if we increases,
Okay.
Temperature increases, photosynthesis increases.
Right. Okay. And what happens if we increase even more? We heat the leaf a bit more.
Also increases.
It increases. So let us say the air temperature in the room is about 25 degrees in your lab. What if we make the temperature 45 degrees? Will the photosynthesis continue to happen at the same rate?
No, sir.
What if we increase it to 60 degrees?
No, sir.
No.
What happens then? What do you think will happen? It will come down, right?
Dry up.
Yeah, I mean, of course, drying is the next part. But if we have a leaf in a chamber like this, that we see in this chamber, let us say we can control the temperature of this chamber. So we can go from 20 degree, 25 degree, 30, 35, 40, 45, 50 degree. So this instrument has facilities to heat the leaf, the air, and through air it can heat the leaf. What would be the shape of that, the rate of photosynthesis? Anybody?
Decreases L shape.
But let us say we reduce the temperature to 10 degree, very cold temperature. From 10 degree to 20 degree, what will happen to the photosynthesis rate?
With the lagging phase of photosynthesis.
It will respond in a way which I will jump the slides and show you this here. So this is an example of one of the trees that we measured in Sirsi, Uttar Kannada district. Okay, and you can see that when we increase each of these the leaf temperature point, okay, leaf at different temperature, and what you can see is when you increase the temperature, photosynthesis increases. There is an optimum point, and after that optima, the rates decline. The rates reduce. So this is a typical temperature response curve. Okay, and here the temperature at which the rate is highest is called the thermal maxima of CO2 assimilation rate or CO2 capture rate. Okay, so this is T. Why is this happening? Why is this a bell-shaped curve? Any idea?
So photosynthesis rate increasing due to the temperature level.
Yes, but why?
Optimum temperatures necessary for.
They require specific amount of the temperature for photosynthesis.
Excellent.
Whether they
I want to hold on to that answer. I can't see who's that, but when you say specific temperature, specific energy, it is the activation energy that we talk about. Activation of which component of photosynthesis are we talking about?
photosystem one
No, photosystem one. Let's go back to this. I'm really happy you're responding. I'm glad. Keep it coming. But look at this slide here. Where are we looking at carbon dioxide uptake?
Rubisco enzymes.
Rubisco is an enzyme, right? Rubisco and associated enzymes. There are a lot more enzymes which are required for carbon dioxide assimilation. But simplifying it for Rubisco and Rubisco activase the enzymes. Enzymes are temperature sensitive. You know that, right?
Yes,
Right? So if you have low energy, low temperature, low activation energy, the enzyme activity will be low. And as you increase the temperature, enzyme activity will increase and then it will decline. So this is your typical enzyme temperature response function. I'm oversimplifying this again because there are a lot of other parameters. For example, stomatal responses. Do you know what are stomates?
it's transpiration.
Stomates. What are stomates? Stomata, stomates. What are those?
Stomata.
Huh. So they're just holes on the leaves, right? On the epidermis, you'll find holes, small holes, through which gas exchange can happen. Stomata, can you hear me?
Yes, sir. Yes, sir.
Right. So through the stomates, when you increase the temperature, when you suddenly change light, when there are different stressful situations, stomates close. That can also affect photosynthesis rate. Okay, so this shape of the curve that we're talking about right now, that I just showed, are independent of that. So we are considering that stomatal dynamics are not affecting this curve. Okay, so let us say if you have a pure extraction of Rubisco and components that are doing photosynthesis, binding with RUBP, absorbing carbon dioxide, and then generating C3 components. If you measure it under an ideal condition, you will find a very clear peak curve that will show you the temperature of photosynthesis of that species, right? But in the natural condition, all of this, the light conditions, the temperature, the humidity in the air, because carbon dioxide is going into the stomates or into the leaf by exchanging water, right? So water is getting released, you're getting carbon dioxide inside, so there is a trade-off, there is an exchange. So if that stops, photosynthesis stops. So there are a lot of components that will affect it. Okay, so this instrument can allow you to change all of this. So this little head in the console, you can say, okay, set the light level to 0, which is switched off. So it can create a dark condition. You can set the light condition to 100, 200, 1000 micromoles. So during a peak day in Chikmagalur, you will have 1000, 1500 micromoles during summer, open sky. So that's the total amount of light that you're getting from the sun. You can change the leaf temperature to a range plus or minus 10 degrees from the ambient. You can change carbon dioxide concentration in this instrument. You can also change water vapor in this instrument. How do we do that? Any idea? How can we change all of this in this instrument? Light level, you can set the LED to different voltage or current level. You can change the light, right? Is that reasonable? temperature, you can heat. You can have plate units, you can heat the leaf or the air. You can increase the temperature. You can also cool it similarly. Now, how will you change carbon dioxide in the atmosphere? So to do that, this instrument, so this is the front side, but on the back side, you will find a few reagents, few components that you need to run this instrument. Okay, and you can see 1, 2, 3, 4 components here. Okay, so it takes the air from the atmosphere, it passes it to CO2 scrubber. CO2 scrubber is soda lime. You pass your, if you remember your chemistry classes, you pass carbon dioxide through soda lime, it absorbs, it binds with carbon dioxide. It removes all the carbon dioxide in the air coming in. Okay. It also removes all the moisture, water vapor, or humidity in the air. So it is removed completely. Okay. Sorry, this is the desiccant I'm talking about. But then you want to set it to a fixed level. You want to say, okay, I want to keep the carbon dioxide to 420 ppm constant because I want to see the response of temperature or light. Okay. So by using the CO2 injector, you can use a small carbon dioxide cylinder. I can show you one of this here. So this, I don't know if you can see my video, but this is a carbon dioxide cylinder. You see this? Yeah. So you can use this in the instrument. This is just liquid carbon dioxide. Okay. And then when you open it, it comes out as gaseous form. And you can add a fixed amount to this flow so that you can control the carbon dioxide concentration. Okay. Similarly, you can remove moisture and then add moisture based on what you need. So the instrument already controls this. You can see its control valves, which are inside. You can set, okay, I want 50% humidity, 420% ppm carbon dioxide. The instrument will set that. In the olden days, for example, the previous generation, I will go back to this picture. Again, you see this little thing here on the top. That is your carbon dioxide cylinder area, which is stained. But here you can see probably a blue or a purple cylinder, right? So this is either soda lime or a desiccant. You have to change the knob manually to absorb or add more carbon dioxide or water into the system. But in new systems, you can already set what you want, and it will give you the desired humidity level, carbon dioxide. Okay, so do we understand this now? We have this photosynthesis analyzer. It is called an ERGA because it's an infrared gas analyzer. You also see people calling it by dual channel ERGA because there are two ERGA, reference and sample, and they compare the two. Okay. And this is a portable system. You can carry it wherever you want. You can connect it to the battery. You can run measurements in the field, in the forest, or wherever you are. Okay. And you can change the leaf environmental parameters, is light level, temperature, carbon dioxide, water vapor. Is that okay? So do we understand this now? So let's go to the forest. Let's go how we measure photosynthesis in the forest. And I think we are already quite almost 1 hour into the webinar. So I'll try and wrap this up quite soon, but moving forward, these are more illustrative pictures. So it will not be difficult. So how do we measure photosynthesis in these instruments? We take the leaf, we clip it in the ERGA, okay? But can we take this up the canopy? How tall is your, for example, banyan tree? Or, hello, can you hear me? Can the audience hear me?
Yeah, I think 7.5 network is sensitive.
Okay. So I will just, should I continue or wait for a couple of seconds?
I think you need to wait. Which for the students, right?
Sure. Okay, let's wait for a second. Sorry about that. And let me just see how many audience we have online. So we have 43 participants. So I really thank all you guys for patience, but I think the next few slides, I have not more than 5-6 slides. These will give you a little more flavor of the measurements in the Western cards. Thank you everybody for signing up and coming in. So this was meant to be a simple webinar. There will be more webinars as we go. There will be more technical webinars that we will do. Yeah, I'm just hoping that we get the connection back in the seminar hall. Do we have any questions from the audience until we have? I know this is quite a simple topic for probably the advanced audience we have, but if there are any questions, so please feel free to put it in the chat. I hope people can actually respond on the webinar chat. I don't know. Do you have the connection back in the seminar hall? Shall we continue? I don't know if it's my network, which is bad. Can someone from the audience okay, so I think now I can see the seminar hall coming back. So can you guys hear me? Okay, so okay, so I can hear. Okay, so we need to mute any other devices in the seminar hall so that we don't get that feedback. Okay, shall we get started? Shall we continue? So there are maximum 5-6 slides, and then these are all images. Okay, so now can you guys see this image? What do you see? The students in the hall? What do you see in this slide?
Sure. Garden around the plant or plant material observed. He was measuring the.
Yes, so we have our youngest scientist, Ashwata, there. I think he's hardly 6 years old. So this was meant to say that anybody can learn photosynthesis measurement. It's not complicated. And I'm not joking. He did pick up quite a lot of how to use this instrument. Okay. So one way of doing measurements in the forest, I'm going to show you 3 different approaches and tell you the advantages and disadvantages. Okay. One most commonly used approach to understand trees is that you cut the branches from the tree. Usually you cut the sunlit top canopy branches, okay? You bring them down and put the cut end underwater, okay? So these branches should be long enough so that the vessels are not cut in the middle. So you need to have longer branches than the vessel length so that you don't get air spaces, okay? And then you cut again underwater so that you clean everything off and then let it take water from the bucket. And then you clip it into this instrument and measure photosynthesis. But what do you think would be the problem with this kind of method? This is easy. So we can bring the branch down to the ground and the kid can measure it. Good. But what are the problems with this?
The plant may respond or not respond for the treatment what you give.
Exactly. That's true. And there are different studies that have actually shown that the photosynthesis rate and stomatal conductance can go down to 80% of the in-tree rate. So if the rates are 10 micromoles on a tree, you will find 2 micromoles rate when you are measuring like this. Okay, so these are very limiting, and sometimes some trees respond quite well. Like you will find the same rate on the tree and on the cut branch. But there's no real way of measuring. You have the entire forest to measure hundreds of species. How will you know that which species is able to maintain the same rate as it is in the tree itself? Okay, so this comes with a huge assumption and is quite limiting. Okay, but people use it. A lot of studies that you find in the literature are using these cut branch measurements. So when you see these studies, you should be careful that they will have the artifacts of branch excision. Okay, what is the other way of measuring it? We just measure where the plants are, where the trees are. I've shown you pictures from 3 different locations. This one is in Sirsi. You can put your instrument on a tripod, maybe create a little platform, measure it on the tree itself. So this is in Sirsi in the Western Ghats. This is my student Philip doing measurement in Uppsala here in Sweden. And this is another student measuring photosynthesis in Puerto Rico. So you can see that we have just clipped the instrument onto the same level as you can reach, right? So in this situation, we are limited by height at which we measure the photosynthesis, the layer of canopy that we can measure. Okay. How do we measure on top of the canopy? Can we measure on top of the canopy without cutting the branches? Any crazy ideas? Come on, I'm sure you guys have thoughts about how to measure. If I ask you guys to measure photosynthesis on a coconut tree without cutting the coconut tree, how will you measure it?
By climbing over it.
You will climb with this instrument. Look at how Philip is carrying all of this on a tripod, on a trolley.
So, but we can tie that instrument to the branches, right?
We can use crane.
Yes, I like the answer of crane. You can use a crane. Yes. What else can you use?
By tying that instrument to the branches.
That's one possibility, but it's very difficult because, as you can see in the picture, they have to be absolutely constant. They cannot move. It will start leaking. So that part has to be static. It's very sensitive to these changes. So I like one of the answers. So what people do is they do this. Okay, so they build these are called towers, right? So these are not mobile towers, but just they're called canopy access towers. There are different kinds, but what you see, this is from Puerto Rico, one of the experiments that is where they're heating the forest to understand the response of forest to increasing temperatures. But you can see that you can actually climb on top of this. You see the stairs here, right? You can go on top, you can measure different layers of the canopy. But how many trees can you measure from this tower?
Drones.
How many?
ಆ ಟವರ್ ಮೂಲಕ ಹೆಂಗ್ ಮೆಜರ್ ಮಾಡ್ತಾರೆ
ಎಷ್ಟು ಮರಗಳನ್ನ ಮೆಷರ್ ಮಾಡಬಹುದು ಈ ಟವರ್ ಇದ್ದಾಗ? Couple of them, right? So you can, if there is one tower, there will be a branch here, another tree here, another tree here, randomly hanging, right? So you can only reach those from that area. Okay, so I don't know if the connection is in the all this back. Oh. Okay, can you guys hear me again? Okay, so okay, so come on, big answer, Madi. I think I have like not more than 4 slides. How many trees can we reach? ಎಷ್ಟು ಮರಗಳನ್ನ ಇದರ ಮೂಲಕ ನಾವು ತಲುಪಬಹುದು?
4 to 5 trees.
4 to 5 trees. But when we measure for one species, we need at least 3 replicate individual trees, alva? When you do experiments, you need replications of it. So sometimes more, because photosynthesis and the parameters are very variable across leaf, across individuals. So to get a species value, you need to measure more trees. So these are beneficial, but are of limited number of trees access. Somebody else said cranes. Can you see the slide? So this is a canopy access crane in Smithsonian Tropical Research Institute, Panama. So you have your crane can travel around, right, 360 degrees, and there is this little hanging basket, and this basket can go up and down. So within an area of 20 to 30 meters, they can drop you in the middle of any tree, and you can measure, like these guys are doing. You see the same instrument. You can measure photosynthesis, right? So there are three approaches, as we just saw. One of them is cut branch measurement, very easy. You just cut the branch, bring it down, has limitations. The other is you can measure it in situ as they are, but at a limited height. You can even measure it at canopy access towers, ideally canopy access. But in the world, there are only a few of these. Okay? Is this okay? Shall we go forward? So now let's make it a bit more intense, or a bit more flavorful for also the online audience. Okay, so are the students with me on this? How many of you have been to Western Ghats Forest? Students, have you guys been to the forest? Kaadi ಹೋಗಿದ್ದೀರೇನ್ರಿ?
ಸರ್ ಹೋಗಿದೆ ಸರ್.
ಕಾಡಿಗೆ ಹೋಗಿದ್ದೀರಾ? ಕಾಡಲ್ಲಿ ಫೋಟೋಸಿಂಥಸಿಸ್ ಮಾಡಿದ್ದೀರಾ? ಓಕೆ, ಸೊ ಮಾಡಿ ನೆಕ್ಸ್ಟ್ ನಿಮ್ ಚಿಕ್ಕಮಂಗ್ಳೂರು ಕಾಡಲ್ಲಿ ಹೋಗಿ ಮಾಡಿ ಎಷ್ಟಿದೆ ಏನಿದೆ. ಸೊ ನಾವೇನ್ ಮಾಡಿದ್ವಿ ಅಂದ್ರೆ, ವಾಟ್ ವಿ ಡಿಡ್ ವಾಸ್, ಸೊ this study was done during 2020 and 21 during the peak corona period. We did photosynthesis measurement on a set of tree species in the Western Ghats forest with the help of all these amazing people. I was in Leeds University then. This was in collaboration with ISER Pune. It was a NERC-funded project. Right now, I'm in Uppsala. So all of these guys were involved in this study. So if you look at this landscape, I don't know if Balu is around or if he has joined in, but Balu was a key person who was instrumental in enabling us to measure and do this study, long-term study. This is only a small part of the study that we did there. You see, the landscape has undulating terrain, right? So there are hills, slopes, and valley areas. And in the valley areas, you find arecanut or rice cultivation. But naturally, if you see valley areas, there are different species. If you come towards hilltops, there are different species. On slope, there are different species. So species have different associations of where they prefer or where they grow ideally. Okay, so if we represent our study site from the hilltop, slope to valley areas, you see that different species occupy different locations. The tree density is higher in the valley areas. The tree density slope is a bit less. But then as you go on top of the hills, usually there are grasslands or deciduous tree species. So this is one of the different formations that you find in this Western Ghats, Central Western Ghats area. There are many such formations that you will find. And these were very meticulously documented by Pascal and others, by the French Research Institute Pondicherry, and Ayyappan and Ramesh and others are actually studying these different formations and different forests across the Western Ghats for several decades now. So into this mix, in this study site in Sirsi, we established a long-term study for about 3 to 4 years. And you know that you get most of your rains during your monsoon period, June, July, August, September, right? So you see the vertical bars, and after that rainfall decreases. If you see this red line here, This is the maximum temperature. Temperatures are low during this post-monsoon period, but then they become high during the dry summer period. You see that the low and high temperature differences are quite stark here. And this is very dry. Okay. If you look at the soil moisture, so you remember these different positions here. So this is hilltop, slope, and valley areas. Okay. So this is hilltop, slope, and valley areas. You see that valley areas have a lot more water than slopes, than hilltops, right? But if you see the two periods, two measurement period, just after monsoon, the water is higher and then it becomes low, right? But it decreases to different levels in different topographic positions. And if you see these and these, this is the graph you get. So the blue is for the wet period, the red is for the dry period. Okay. Generally, wet period has more water, dry period has less water. Fair enough. Okay. So, but the difference is that the hilltops experience is very small compared to the valley areas, for example. Okay. So this is just the surface water. And what this tells us is that within a small area of just a 50-meter vertical topographic difference and about 50 to 100-meter lateral change, you can find different species, different soil moisture levels, and even different microclimate conditions. So different trees are at different thermal environments, if you want to think of it. We measured temperature sensitivity of photosynthesis on all of these species. So now you remember these are the three hilltop species, these are the slope species, these are the valley species. Okay. And the blue color temperature response curve are for just after the monsoon period. So let's call it as the wet period. Okay. The red ones are during the dry period or the summer period. Okay. And this is how the curves look like. But to understand this better, what we can look at is the figure on the right hand side. Okay. So these represents the A opt or the maximum photosynthesis rate at the thermal optima. Okay. So let's look at these. So these are different species. These are the hilltop, slope, and valley species. What do you see among these three species, the red and blue? Do you think they are different? Seasonal difference? Do you see any seasonal difference here? In these three species?
Slight difference.
Very small difference, and they are not even statistically significant. So if you see these, they have the same photosynthesis rate between wet period and a dry period. That means they have water, they have low rates, but they maintain their rates as they are. Let's look at the hilltop. These are all deciduous, these three species. During the dry period, their photosynthesis is low. They have less water, for example, they have more temperature, the photosynthesis goes down. But during wet period, the photosynthesis rates are higher. Good. Do we understand it? Yeah. The surprising aspect for us was the slope species. Okay. The slope species showed lower photosynthesis rate during the wet period, whereas they showed higher photosynthetic rate during the dry. They preferred a warmer thermal environment to maximize the photosynthesis rate between the two seasons. And this study is under review in Physiology of Plantarum. This is also openly available to the preprint read. So this was the study we did in the Western Ghats forest. And I represent that here in a similar way. So what you see here is the AOPT aggregated for all the three species together, right? And the soil moisture differences plotted against them. So this was a study. Are you guys with me now? So I think we have already passed 1 more minute beyond the set time period. So it's quite late. Do you want to know about a bit of an Amazonian study as well? I have one slide.
Sure.
So Amazonian forest. This is just literally one slide. I can send the link to your teachers and you can read this. This entire paper is also in Kannada, so you guys can read it in Kannada if you want. Okay. Now, you guys know that Amazonian forest is the largest terrestrial carbon pool on this planet. So if this entire northern part of the Southern America is covered with Amazonian forest and many other forests, but Amazon is the largest. And there we went to the warmest site possible, right? So Chikmagalur is a bit cooler site. So let us say if you go to a bit more warmer site in Karnataka, let's say a bit more northern side, the temperatures will be higher, right? Similarly, we went to this site, which has the highest air temperature during the dry period, and we measured heat sensitivity of photosystem 2. So this is light reaction. So we were talking about dark reaction, carbon dioxide assimilation rate until now. Now we are talking about the light reaction and photosystem 2 heat sensitivity. Okay. What you see here, just look at this line, the sigmoidal curve. This represents the integrity of photosystem. At 30 degree, it is fine. At 35, it is fine. 40, it starts decline. At 50 degree, it is half. At 60 degree, it's almost zero. Right? So structurally, it has broken down, or functionally, it has broken down. And this is a measure which we usually refer to as thermal tolerance. How do we measure it? We take the branches as is shown here. We take the leaf, we treat them to different temperature for a fixed period. We measure the fluorescence of chlorophyll fluorescence of photosystem 2, and that is what is plotted in this axis here, y-axis. Okay, so and what you see here as histograms are the temperature records for that site, and the increase that you clearly see at the temperature at which they are becoming sensitive are now overlapping. So if you just see the white histograms, they were not heating up the photosystems where they are already temperature sensitive, but now they are heating up these systems. So this was done with an university in Mato Grosso, the wonderful friends and team there when I was in Leeds. And I think I will leave it at that. If you can recognize your teacher there, and if you can recognize me. Do you see the slide? I don't know if you guys see the slide. Slide can't stay there. Last slide.
Yes, sir.
Okay, so you can identify your own lecturer there, and if you can find me. Okay, good. So you can scan the QR, and maybe we can take a few questions. And if Ganeshji wants to coordinate a little bit. So sorry about the slow pace of the presentation, but I hope you guys got something out of it.
Questions is like it. Because now it is open to even other participants from other institutions can also ask questions.
Maybe shall we open this up for online audience? Maybe. I've just asked unmute. I don't know if that's going to be chaotic, but I request audience if you have any questions, if you want to look at any of the.
So what is the duration of the instrument? ಎಷ್ಟೊತ್ತು ಪ್ಲಾಂಟ್ ಇರಬೇಕು ಅಂತ ಲೀಫ್ ಅಲ್ಲಿ ಕ್ಯಾಪ್ಚರ್ ಮಾಡಬೇಕು ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್?
Excellent question. So if you see the changes in temperature through the day, changes in light through the day, every hour temperature is changing, every hour light is changing, right? So if you keep the leaf in a single situation for too long, it is not natural. So typically what we do is we clip the leaf in the chamber, we change things within minutes, couple of minutes, do our entire curve. If light response curve is like 10 to 15 minutes, carbon dioxide response curve 10 to 15 minutes. Temperature response curves take like 1 hour, 1.5 hours, but we try and do it as short as possible because we should not be changing the entire aspect as well. We shouldn't be conflating the results. Does it answer your question?
Yeah, okay, sir.
Yeah. Any questions? Any more?
Sure, we have the carbon dioxide tube for maintaining the concentration of carbon dioxide. So there is why need of carbon dioxide scrubber and the inlet of air.
Excellent question. Excellent question again. So let's go to that slide. This one, right?
Yes.
Now,
Next
in
one.
the room that you have, right? So this slide.
Yes.
Okay. So in the room, all of you guys are respiring. You're releasing more carbon dioxide, right? Carbon dioxide concentration in that room will be very high. If you ventilate the room, if you open it up, if you turn on the fan, it will come close to 400, 420 if it's empty, if it's all well ventilated, right? If you go to different locations, for example, carbon dioxide concentration keeps changing. So if your input gas is changing, how will you know what is in reference and what is in the sample? Number one, does it make sense? So we want to know what is exactly coming here and how much of it is getting removed. Okay. And we don't want a leaf to be responding to carbon dioxide change. So if you keep a leaf in your office, in your lab, it will see more carbon dioxide than it is seeing outside in the forest, in the open area. So we don't want that. And if you expose yourself or there are more people in that room for a long period, there will be a lot of carbon dioxide and people will start sleeping because carbon dioxide will make people dull in that room. That's why if you have long sessions of classroom, you should also go out, ventilate, open the windows, put on the fans, or else you will all sleep without actually intending to sleep. It's carbon dioxide that is making it slow. So, and in a very simple way, if you want to measure something, you have to keep that constant. Right? So when you if you want to measure your weight, you will get on the scale, right? But will you keep jumping on and off of it or keep adding your friend to it? You have to keep that constant, right? That is the idea. So whatever components can affect your values, you have to keep it constant and then measure the change, which is the result of leaf. Any more questions?
So, what is the tolerance of thermal tolerance in the in dual channel, sir?
Okay, you're asking thermal tolerance in
Dual channel. The leaf, the tolerance.
Okay, so these are two different techniques. Okay, so let's say whatever I described here only has a small little instrument. This doesn't have gas exchange measurement, doesn't give you photosynthesis rate. It only gives you if the photosystem 2 is functional or not, if it's fluorescing or not. It doesn't give you carbon dioxide. This is a simple instrument. You see this little thing here, tiny instrument, or I can show you others. For example, like these. So the one on the left-hand side, they probe photosystem 2. The one on the right-hand side, something like these, will probe gas exchange, like this. Okay. And any more questions?
ಸರ್ ಇನ್ ಕಾನ್ಸೆಪ್ಟ್ ಕ್ಯಾಚ್ ಮಾಡಬೇಕಾಗಿಲ್ಲ ಸರ್. ಅಂದ್ರೆ ಎಲಿವೇಶನ್ ಜಾಗದಲ್ಲಿ ವಾಟರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಅಬ್ಸರ್ಪ್ಷನ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ. ಏನ್ ಸರ್ ಅದು?
Okay, so you were talking about
the mountain valleys range.
Yes, here. So if you think of water, ಇಲ್ಲಿ ನೀರಿದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ, ಮಳೆ ಬರ್ತಾ ಇದೆ ಎಲ್ಲ ಕಡೆ. ಮಳೆ ನೀರು ಯಾವ ಕಡೆ ಹೋಗುತ್ತೆ ಜನರಲಿ? ಯೂಶಲಿ downwards ಹೋಗುತ್ತೆ. So ಇಲ್ಲಿ you will find more water. So the water levels will be higher in the surface. But there's someone who wants to speak, but yeah, maybe. But in the hilltops, they get percolated more quickly. There is less vegetation. They hold less water. But generally, you will find more dry affinity spaces here, and then in between, you'll find different water affinities. So I'm trying to see if I can. There's someone who wants to speak. How do I see this? Okay, so there's someone raising the hand, I suppose, and how do I? Yeah, if you guys, whoever is raising the hand can actually speak. Oh, okay, I see question and answer session. Okay, sorry, good Swapna. Okay. So I'll probably just take the questions right now. If water level is more in slow areas, does photosynthesis is less compared to more slow regions? I'm not sure. So Vishwas, if you want to actually ask your question, I think I've asked everybody to unmute, so you can actually just ask your question directly. Attendees, I'm not sure if I understood the question, so feel free to just ask me a question directly.
Same question. Same question.
ಸರ್ ಇದು ಸ್ಲೋಪ್ ಇರೋದ್ರಿಂದ ಸರ್ ಆ ಲೈಟ್ ಇದೆಯಲ್ಲ ಸರ್ ಲೈಟ್ ಅಬಿಲಿಟಿ ಮೇಲೆ ಜಾಸ್ತಿ ಇರೋದ್ರಿಂದ ಸ್ಲೋಪ್ ಅಲ್ಲಿ ಸರ್ ವಾಟರ್ ಇರೋದ್ರಿಂದ ಅದ್ರದ್ದೇನಾದ್ರು ಲೈಟ್ ಇದೆ ಸರ್ ಫೋಟೋಸಿಂಥಸಿಸ್ ಏನಾದ್ರು ರೇಟ್ ಏನಾದ್ರು ಕಮ್ಮಿ ಆಗುತ್ತಾ?
Okay, so your question is, if light availability changes to these different points, will that affect photosynthesis? Excellent question. We measured light levels in all of these, and then we tested if that explained the photosynthesis variation that we measured. There was no relationship. You would expect if there is more light, there will be more photosynthesis, but different species and at different canopy levels are built for different levels of light saturation. So they can only use certain amount of light. So probably that is masking the effect at this aggregate level. Does that answer your question? So generally, I'll try in Kannada. So ಇಲ್ಲಿ ಏನಾಗ್ತಾ ಇದೆ ಅಂದ್ರೆ, ನಾವು ಹಿಲ್ ಟಾಪ್ ಸ್ಲೋಪ್ ವ್ಯಾಲಿ ಹೋಗ್ತಾ ಇರ್ಬೇಕಾದ್ರೆ ಇಲ್ಲಿ ಮರಗಳ ಸಾಂದ್ರತೆ ಕಡಿಮೆ ಇರೋದ್ರಿಂದ ಇಲ್ಲಿ ಬೆಳಕು ಹೆಚ್ಚಿರುತ್ತೆ, ಇಲ್ಲಿ ಹೆಚ್ಚು ಮರಗಳ ಸಾಂದ್ರತೆ ಆಗ್ತಾ ಇರೋದ್ರಿಂದ ಇಲ್ಲಿ ಕೆಳಗಡೆ ಅಂಡರ್ ಸ್ಟೋರಿ ಅಥವಾ ಇಲ್ಲೆಲ್ಲ ಬಂದಾಗ ನಿಮ್ಗೆ ಬೆಳಕು ಕಡಿಮೆ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಟಾಪ್ ಆಫ್ ದ ಕ್ಯಾನಪಿ ಸೇಮ್ ಇರುತ್ತೆ, ಎಲ್ಲ ಟಾಪ್ ಆಫ್ ದ ಕ್ಯಾನಪಿ ಸೇಮ್ ಇರುತ್ತೆ, ಬಟ್ at the level where we measured, light levels will be highest here, intermediate, low. Water levels, intermediate, high. Okay, na?
ಸರ್ ನನ್ನ ಪ್ರಕಾರ ಸರ್ ಅದು ಫೋಟೋಸಿಂಥಸಿಸ್ ಕಮ್ಮಿನೇ ಆಗುತ್ತೆ ಅನ್ಕೋತೀನಿ, ಬಿಕಾಸ್ ಸರ್ ಆಲ್ಟಿಟ್ಯೂಡ್ ಮೇಲೆ ಹೋದಾಗ ಟೆಂಪರೇಚರ್ ಲೋ ಆಗುತ್ತಲ್ಲ ಸರ್.
Temperature ಅಷ್ಟಾಗಿ ವ್ಯತ್ಯಾಸ, that's actually a question. We did not measure temperature here, so I don't know if Balu is online. So Balu studies these different topography and microclimate temperature conditions, and they vary. But in this particular study, we did find a variation. Here, the temperature was the highest. This is the intermediate, and this is the lowest air temperature, because this is all sun exposed during the summer period. So
ಫೋಟೋಸಿಂಥಸಿಸ್ ಮೇ ಬಿ ಲೋ ಓವರ್ ದ ಟಾಪ್, ಹೈಯರ್ ಆಲ್ಟಿಟ್ಯೂಡ್ ಕಮ್ಮಿ ಆಗುತ್ತೆ.
This is what we find. For example, let's take, yeah, this is what we find. Let's look at this. So the blue is for the wet period, The red is for the dry period. So if you look at summer period, the first graph in this 3D, so it increases hilltop slow, but then valley is low for the dry period. But for the wet period, hilltops do the maximum photosynthesis, then this one is intermediate, this is low. That's what we find. Okay.
Okay.
Any more questions? Okay, if you guys have phone, you can scan this and then you can respond to that.
Sir,
Huh. Go on, go on.
ಸರ್ ಡೆಸಿಡಿಯಸ್ ಫಾರೆಸ್ಟ್ ಅಲ್ಲಿ ಜಾಸ್ತಿ ಫೋಟೋಸಿಂಥಸಿಸ್ ಆಗುತ್ತೆ ಅಥವಾ ಡೆವಲಪ್ಡ್ ಫಾರೆಸ್ಟ್ ಅಲ್ಲಿ ಫೋಟೋಸಿಂಥಸಿಸ್ ಜಾಸ್ತಿ ಇಲ್ಲ?
Excellent question. Really, where we studied, this is a combination of both, right? So in the same forest, you will find evergreen and deciduous. Okay. The rate of photosynthesis leaf level deciduous will be higher, as you can see here during the wet period. Evergreens are slow growing. But in one location with a mixed species, this is what we find. And if people want to conclude, it's almost 2 hours now, but I'm really glad the online audience are also sticking along. But if there are not any more questions, we can have one more session where we can discuss more about this. But can you guys scan this? And maybe if the organizer wants to conclude, so you guys can scan this and then respond to these questions. These are not your exam questions, it's just to get feedback on how the session was and how we can improve. So you can scan this QR, and if Ganesh or others wants to conclude this. Okay. Shall we conclude? Close Madma.
Yes, sir. Sir, we want to hear from our principal. Yeah, Pushpa Bharati.
I can't hear. I think I muted people. So whoever is speaking, could you unmute yourself and then speak? So if you want to conclude, we can quickly do that.
Sir, I request our principal, Dr. Pushpa Bharati R.I. Madam, to propose what presidential address. Sir, ಅವರಿಗೊಂದು ಅನ್ಮ್ಯೂಟ್ ಆಪ್ಷನ್ ಕೊಡ್ಬೋದು.
So could you sorry, I think sorry, where was this? ಹೆಸರು ತಿಳಿಸ್ತೀರಾ?
Dr. Pushpa Bharati R.I.
Right, I'll talk. Okay, I think, yeah.
Am I audible?
Yes, yes.
Yeah.
Madam, over to you.
I'm not sure if I can hear anyone.
Am I audible now?
Yes, yes.
All respected all dignitaries, participants, and dear students. Today with the IDSG Government College, Chikmagalur, Karnataka, India, have hosted a webinar in coordination with Uppsala University on the topic measuring heat sensitivity of photosynthesis in plants. Our bright resource person, Dr. Rakesh Tiwari, enlightened us in this regards very well. As you know, he shared the knowledge about the topic very well. Our students interacted and discussed the topic with Dr. Tiwari. I think they got answer to their questions and doubts. I hope that is the greatest and ultimate use of this webinar. So I'm very thankful to our resource person, Dr. Rakesh Tiwari. And I want to extend my thanks to Uppsala University also. This webinar went nicely in the form of classroom only. Even I also felt that I'm sitting in a botany class and having the knowledge about. I'm very happy to say this is a success. For this, I congratulate the HOD of Botany Department, PG coordinator, and all lecturers, and also stakeholder students. In future also, we would like to have this type of knowledge exchange programs. With this wish, I will end up my words. Thank you. Thanks one and all.
Thank you, madam. Thank you, madam, for your encouraging words. Sir, shall we wind up?
Yeah. Thank you everyone for the opportunity.
Last words, concluding remarks.
Okay, sure.
Yeah, thank you. Design principles based on scientific understanding of photosynthesis was the key outcome for today's session. Understanding internal and external factors, their effect on photosynthesis at varied degrees bring unique ways of understanding the process at different levels. Carbon dioxide being a curious chemical which plants are capable of handling or sequestering effectively has brought a possibility of measuring heat sensitivity of photosynthesis in plants. This fact has been scholarly presented by Dr. Rakesh Tiwari sir in a very lucid yet concrete manner. Thank you for this, sir. We are also happy for making this webinar on a discussion mode so that our students got a chance to interact with their own thoughts and logics. Thank you for that also, sir. I thank Dr. Rakesh Tiwari sir for providing this wonderful opportunity for us. I thank all the participants from various institutions across 22 states of India and 4 other countries witnessing this webinar. My thanks are due for our principal, Dr. Pushpa Bharati R.A. Madam, for permitting us to conduct this webinar, and appreciate my students and colleagues for their enthusiastic participation. With the hope of more guidance and future collaborations, I conclude today's webinar. Thank you, one and all.
Thank you. Can you close this one now? Yes.